Hello, I am Simone Marini, and this talk is called Learning Antimicrobial Resistance through Secondary Structure of Protein Variants. Antimicrobial resistance, or AMR, is a significant and growing public health problem. There are several methods to automatically identify resistant bacterial strains, yet we are in need for computational methods to predict novel AMR variants. AMR variant is a genomic variant that, if present, will confer the bacteria resistance. The idea from a molecular perspective is that the variant changes the secondary structure, well, in general, the structure of the protein that is encoded, and these alters the way that the protein interacts with its surrounding molecular environment, hence the resistance. As a consequence, we can or we could be able to predict AMR based on the knowledge of the secondary structure or the variation in the secondary structure. We do not know the secondary structure of all the proteins, but we can predict it. And here we used brewery, which is a very recent deep learning method to predict secondary structure. And we focused on relative solvent accessibility, which is the level of exposure of an amino acid to its surrounding molecular environment. The assumption is that, of course, if a variant is providing resistance, probably this variant will have an exposed amino acid, because in order to give the resistance, there needs to be an interaction with the environment. Now, brewery for any amino acid or protein will output for the probability distribution for 4 classes, namely very buried, somewhat exposed, and very exposed. So here we extracted from the CARD database, which is our AMR database, 1,234 pairs of protein variants from 152 proteins. And we measured the score for each variant in each of the classes. Of course, we have pairs of variants, and we have the susceptible variant and the resistant variant. And when we measure the scores, we find that susceptible variants tend to have a higher score for the very buried class, as opposed to resistant variants that tend to have a higher score in the somewhat exposed or very exposed classes, which means that the resistant variants tend to be more exposed, and the susceptible variants tend to be more buried. We also took these 4 scores, and we used them to construct classifier to predict susceptibility or resistance. And although the results are not strong enough to have a machine learning classifier based only on that, we found that there is non-trivial signal there in the relative solvent accessibility to predict resistance or susceptibility, as shown by these ROC curves from logistic regression, J48, and CART decision trees that are distant from the baseline. So we looked further into the CARD dataset, which so far has been analyzed as a whole, but we know that it's composed of different organisms and different AMR mechanisms. So we wanted to verify if our findings were reflected also into specific mechanism and organism combinations. So we selected all the possible subsets made of specific organism and AMR mechanism combinations, and we retained only the ones that had at least 30 pairs of susceptible and resistant variants. And we found 6 of them. We see that in the different subsets, again, the scores for resistant variants in the class that is very buried, they tend to be lower than the susceptible ones, but they tend to be higher for the classes that are somewhat exposed or very exposed. Now, this holds true for 5 out of 6 subsets. Interestingly, for the organism Clostridioides difficile and the resistance to fluoroquinolone, we have a different score distribution. And we see that susceptible variants tend to have a higher score for the somewhat exposed class, and the resistant variants tend to have a higher score for the very buried class. So this will be, it has to be looked further and might depend on the molecular nature of this specific resistance mechanism. In summary, this study is a first step towards an automated mechanism to classify or discover novel AMR protein variants.